Good morning or good evening, everyone. My name is Daniele Piomelli, and I am a professor in the Department of Anatomy and Neurobiology at the University of California, Irvine. I've been tasked today to talk to you about the endogenous cannabinoid or endocannabinoid system, the system that is normally engaged by the active principle in cannabis delta-9 tetrahydrocannabinol, or THC. I'd like to start off by giving you a quick historical overview of the discovery of the endocannabinoid system. And that actually starts with the very discovery of cannabis by modern science. We have a date for that. The year is 1845, and we have a place for that. The place is Paris, France, where a psychiatrist by the name of Jacques Moreau de Tours published a book on cannabis, the first such book in modern medicine, in which he described the properties of the plant and also highlighted some potential therapeutic applications. So we can say that 1845 is the year cannabis was introduced in modern science. Soon after its introduction in the 1850s until the 1940s, cannabis was listed in the pharmacopoeias throughout the world. The pharmacopoeias, of course, are the contain a record of all the agents that can be used in therapy. And cannabis was listed in the US pharmacopoeia, in various European pharmacopoeias, as an analgesic, an antispasmodic agent, and a narcotic. Things changed at the end of the 1930s with the introduction of a piece of legislation called the Marijuana Tax Act in the United States that made cannabis completely illegal. In fact, similar legislations occurred throughout the world, and cannabis became, from being essentially a medicine, an illegal substance of abuse. That slowed down research, but it didn't completely stop it. In fact, between 1944 and 1964, through the effort of various chemists, including Roger Adams in the United States and Rafi Meshulam in Israel, the active principle of cannabis, tetrahydrocannabinol, THC, was discovered, fully characterized in 1964. It took another 20 years for that to lead to an additional scientific advantage. And that slowness, that slow development is partly due to the fact that cannabis continued to be illegal. In fact, now a new piece of legislation, the Controlled Substance Act of 1970, confirmed the legality of cannabis in the United States of America, and similarly throughout the world. Now, even though slowed down, research did not stop, and between 1988 and 1990, the first component of the endogenous cannabinoid system, the cannabinoid receptors, were discovered. In fact, that's the date where the first such receptor, the so-called CB1 receptor, was identified by Eileen Howlett and also by Lisa Matsuda. Eileen Howlett characterized it pharmacologically, and Lisa Matsuda characterized it molecularly. Soon after the discovery of the cannabinoid receptor, flurry of activity to understand how these receptors were normally recruited in the brain and the rest of the body led to the discovery of the endocannabinoid system, which is the main topic of my presentation today. So now, one thing that you may surmise looking at this is that we have worked on cannabis for so many years. We know a lot about it. In fact, we don't, and in large part is because of the illegality that has shrouded this substance in a way that has mostly hurt research, in addition to underrepresented communities, but for science has been really a very slowing factor. And so now we are in 2020 with 43 states of the Union and USA and the District of Columbia having cannabis as a legal medical substance, and we still have a lot of questions about that. So one thing that we do know, I think fairly well, is how cannabis works. In fact, when we say how cannabis works, what we mean is a number of different things, but the most obvious one is how does cannabis produce its intoxication, the high that folks who use it for non-medical purposes seek, is due, I would say almost entirely, to delta-9 THC, the structure of which you can see here. So this is a very complex molecule, different from many other active principles in plants. For example, very different from cocaine, very different from morphine. And in fact, it's not even an alkaloid, as you can see. It is a terpene, very fat-like molecule. And that has some interesting consequences which we will explore briefly later on. But we know that THC works by combining, just like a key combines with a lock, combining with cannabinoid receptors throughout the brain. And so we will see also the rest of the body of humans and other mammals. So I alluded before to the cannabinoid receptor. I mentioned the work of Lynn Howlett and Lisa Matsuda, who identified pharmacologically and molecularly the first of the two known cannabinoid receptors, named, of course, CB1. What you see here are two fairly fanciful structures. This is not necessarily how the receptor looks. They've been generated using computer modeling, but I put them there to highlight the first important property of these two receptors, that although they bind THC fairly equally, they're quite different in their molecular structure. In fact, they only share about 44% sequence homology. Not only are they different in structure, but they're also different in localization. The CB1 receptor is a very abundant and ubiquitous receptor. Ubiquitous, present essentially in every single organ and tissue of the body, with few exceptions. It is extremely abundant in the brain, but it's also present in peripheral neurons, the population of neurons that live outside the central nervous system. But surprisingly, it's also present in cell lineages that we don't really think about when we think about the effects of cannabis. And I'm talking about cells like the adipocytes, which are the fat cells in white and brown adipose organ. The hepatocytes, the cell that constitute the large majority of the structure of the liver. So CB1, very abundant in the brain, but also ubiquitous. What about CB2? CB2 is a lot more restricted. It is actually present in high levels only in cells of the innate and adaptive immune system. For example, macrophages, part of the innate immune system, or B lymphocytes, part of the adaptive immune system. And it's present at lower levels also elsewhere. There is some in the brain. It's still unclear whether it's present only in the innate immune cells of the brain, the microglia, or also in neurons, but clearly one can find it in the brain as well. One thing that I like to point out is that even though the CB2 receptor is much more restricted in the type of cells in which it is expressed, those cells can be very widespread. For example, the macrophages are present in every organ system of the body. Therefore, if they carry CB2 receptors, we will have CB2 receptors in, say, the skin, and in liver, et cetera, lungs, et cetera. So when we talk about cannabinoid receptors, we're talking about a receptor system that is really widespread and quite significant. Now, for the aficionados, the pharmacologists in the audience, maybe not very many, but for the aficionados, I really need to point out that these two receptors are coupled. They normally sit in the membrane, of course, of cells, and they're coupled to intracellular signaling mechanisms through G proteins, which are specialized proteins that bind to the receptor, but also they bind to other proteins that mediate the effects of receptor activation. For example, enzymes that regulate cyclic AMP levels, calcium channels, potassium channels, protein kinases. In fact, both CB1 and CB2 receptors have a really rich complement of intracellular transduction mechanisms that mediate the differential effects in different cell types. So this is a schematics showing the localization of CB1 receptors in the human brain. So the denser the expression, the darker the color. So you can see here, for example, in the cerebellum, very high density of the receptor, also in the basal ganglia. cerebellum and basal ganglia, two very important regions for movement. They coordinate movement initiation, and the presence of cannabinoid receptors in these areas suggests a very important role for the endogenous cannabinoid system in the regulation of those functions. But you can see that there is a receptor pretty much everywhere, for example, in regions like the amygdala, which is involved in anxiety and fear, or the hippocampus, which is involved in memory. Also worth mentioning is the fact that receptors, CB1 receptors, are not very abundant in some other regions of the brain. And I like to point out, for example, this whole region called the brainstem. And I like also to point out this specific nucleus here, or complex of nuclei, called the Pre-Bötzinger complex, which regulates respiration. The Pre-Bötzinger complex regulates the mechanics of respiration, so the rhythm of respiration. And there aren't very many cannabinoid receptors in that region of the brain. And that's important because it also explains why cannabis is not a very toxic substance, because substances like morphine or heroin, chemicals that interact with receptors that are present in the Pre-Bötzinger complex, there are opioid receptors here, tend to cause respiratory depression or even suppression. And this is something that the cannabinoids, THC in cannabis, doesn't do. So this is CB1 and the central nervous system. Let's take a look now outside the brain. Outside the brain, as I said before, there are at least two subtypes of cannabinoid receptors. And very surprisingly, when these discoveries were made in the 1990s and early 2000s, very surprisingly, these receptors are present everywhere. I cannot overemphasize that, but if you just take a look at this slide, you will see how many different functions of the body are potentially regulated by cannabinoid receptors expressed in different organ structures. I would like to point out, for example, the presence of cannabinoid receptors on peripheral nerve terminals. Okay, peripheral nerve terminals are the terminals of pain sensing or mechanosensitive neurons, the cell bodies of which are not located in the CNS. They are located outside the CNS in structures called the dorsal root ganglia. And CB1 receptors are very abundant in a subset of neurons present in the dorsal root ganglia, particularly some of the neurons that transmit the pain signal from the periphery of the body, say from the skin or the mucosae, to the spinal cord and the rest of the central nervous system. That's surprising because initially people thought that cannabinoid receptors involved in regulation of pain or elsewhere would be just in the brain. But no, they're also in the periphery. Another interesting localization for the CB1 receptor is the small intestine. And the small intestine is not a place where you associate with cannabis, right? Just like you don't associate cannabis with, say, the lungs or blood vessels. But in the small intestine, the effects are not just what you would expect, the regulation of small intestine motility, which does occur, but also regulation, for example, of hunger. It has been shown that cannabinoid receptors in the small intestine regulate through a complex indirect mechanism hunger. So we're looking at really a very rich, complex, and widespread signaling system that hinges on these cannabinoid CB1 and CB2 receptors. Why do we have cannabinoid receptors? True, humans have used cannabis for approximately 11,000 years. We have good archaeological evidence that there was cannabis cultivation in Europe, Japan, and various parts of Asia, at least 11,000 years ago. It's likely that some of that was for religious purposes and so involved intoxication. However, that kind of time span is not enough to generate receptors due to a coevolution. Clearly, what cannabis does, it hijacks a system that we have and we had for probably hundreds of thousands of years, and if not millions of years, as mammals. And that's the endogenous cannabinoid system or endocannabinoid system. So the endocannabinoids are the chemicals produced by the body that activate normally cannabinoid receptors in the brain, as shown here in this particular slide, but also elsewhere in the body. Now, the receptors that are activated by endocannabinoids in the brain are, as you may remember from one of my previous slides, located in a variety of regions of the brain. So it's not such a big surprise that a variety of functions of human behavior are regulated by endocannabinoids. And I'll start off with feeding. That is perhaps the most intuitive one, because anyone who knows anything about cannabis knows about the munchies, that experience of being hungry after having consumed some cannabis that is due to the fact that cannabinoid receptors are localized probably in regions like the hippocampus and the intestine that regulate hunger. Emotion is another very, very important function regulated by the endocannabinoid system. We know this from hundreds, if not thousands now, of animal studies, but even human studies. And anxiety and depression are two important areas in which the endocannabinoid system plays a modulatory role. Pain and memory are also involved, are also regulated. Pain in a positive way, if I may use this expression, that is, endocannabinoids tend to have analgesic or pain-killing effects. Memory is in a perhaps not so positive way, because the endocannabinoid system tends to actually reduce certain forms of memory. For example, the memory for traumatic events tends to be erased thanks to the activity of the endocannabinoid system. That is a good thing, but you can also lose other memories that aren't probably useful. So the effects of cannabinoids on memory are kind of mixed. And finally, reward. By reward, what I mean is that the drive, the emotional drive to, and the pleasure that one experiences in life, through activities such as sexual activity or eating or other forms of pleasurable activity, the endocannabinoid system plays an important role in that. So who are the characters of this endocannabinoid system? There are two molecules that have been, over the years, fairly well characterized as the key component of the system. They're not the only ones, but they are those that I think matter most, at least based on what we know now. One is called anandamide, and the other is called 2-AG. 2-AG is an abbreviation, of course, for a much longer chemical name, which is 2-arachidonyl glycerol. It's pretty much of a mouthful and not as well-sounding as anandamide. So, but this is the name that we have selected, and this is the name that we will be using. So anandamide and 2-AG. What is special about these compounds? They are really, really special. And what is special about these compounds is that, unlike other transmitters, they are not hydrophilic, water-soluble molecules that are normally stored in vesicles and are released when there is an action potential that triggers electrical activity, etc. That is not what happens. The anandamide and 2-AG, the endocannabinoids, are actually produced, they're made upon demand. Before need arises, they're not there. They are present in a precursor form in the membrane of a number of cells in the body, probably most cells in the body, and they're produced only when need arises. So again, that's different from other known neurotransmitters like serotonin or glutamate in a fundamental way. Like serotonin or glutamate, however, anandamide 2-AG must also be rapidly destroyed. So this is something that they have in common with other transmitters. Anandamide and 2-AG both bind to and activate the cannabinoid receptors, CB1 and CB2. They have their preferences, but by and large, they both activate CB1 and CB2. But despite this, they're functionally different in subtle ways. So 2-AG, let me start with this. 2-AG is really the workhorse of the endocannabinoid system in the brain. There is a lot of 2-AG, a lot of that 2-AG is involved in endocannabinoid signaling, and in particular is involved in a phenomenon we will describe briefly in a couple of minutes called retrograde signaling, a form of point-to-point signaling that I will give you a description of later. What about anandamide? Anandamide is more of a traditional modulatory transmitter. So a transmitter that is produced in a certain point of the brain or elsewhere and acts within a volume, a certain distance from its site of production, and modulates the activity of other transmitters. Again, I'll give you a more specific definition and some examples of that in a couple of minutes. Because of the role of 2-AG as a point-to-point retrograde messenger, this molecule plays multiple roles in the CNS and in the periphery. Way too many for me even to touch upon briefly today, but I will go back and describe you the phenomenon of retrograde signaling. What about anandamide? Anandamide is more specialized in the central nervous system. The main roles that have been identified for anandamide are regulation of social behavior and the stress response. And by stress response, I want to go back to what I said before about regulation of emotion and also regulation of pain, because of course a lot of our emotions derive from the exposure to stressful events happening outside ourselves. So in the CNS, social behavior and stress response. In the periphery of the body, instead, the main role of anandamide is to regulate the entry of meaningful, painful events from the periphery into the CNS. So let me explain briefly what is this retrograde signaling concept. For those of you who don't do this every day, well, so this is a synapse, and the synapse is the place where neurons talk to one another. So there is a presynaptic component that comes from one cell, and there is a postsynaptic component that belongs to a second cell. This presynaptic cell signals to the postsynaptic cell, tells the postsynaptic cell be active or don't be active. So how does it do that? It does that through chemical transmission. Chemical transmission operates as follows. There are tiny little vesicles in the presynaptic bouton, as it is called, and these vesicles contain molecules of a transmitter. For example, glutamate, an excitatory transmitter, or GABA, gamma-aminobutyric acid, an inhibitory transmitter. When an electric stimulus arrives in the presynaptic bouton, this is called an action potential, calcium enters the presynaptic bouton, and the calcium inside the presynaptic bouton is able to trigger the phenomenon of neurosecretion, and now these vesicles fuse with the membrane of the presynaptic bouton, go outside. Now they discharge their content outside, and the content, you can see these little blue spheres, could be, say, glutamate, for example, or GABA. Let's say it's glutamate for this particular case. And glutamate binds to receptors. They're not shown here, but they're present in this region, and these receptors mediate the effects of glutamate. So glutamate is excitatory, as I said before, so receptors will bind the glutamate, will open up, and calcium and sodium, all these interesting ions, will flow through, and the postsynaptic cell now will become excited. So this is a classical phenomenon of a description of anterograde transmission at an excitatory synapse. Now, the way endocannabinoids work, in particular the way 2-AG works, is that they start operating when classical transmission is over. Basically, what happens is that in the case of glutamate, for example, glutamate will bind to a receptor, a modulatory receptor. In this case, it's called the metabotropic glutamate receptor. There are several types of this. This could be mGluR5 or mGluR1. And this receptor, upon binding glutamate, engages a system of enzymes that are located nearby. So these three little symbols here represent the glutamate receptor and two distinct enzymes, one of which is called diacylglycerol-lipase, or DGL. The other is called phospholipase C, but that doesn't really matter. What I think it's important is that these three actors are all linked together. They're literally linked physically together by scaffolding proteins. And when glutamate arrives, activates the mGluR receptor, the mGluR receptor transmits this information to the enzymes close to it. And now these enzymes start cleaving, start hydrolyzing the precursor for 2-AG, which is another lipid that normally sits in the membrane, in this particular part of the postsynaptic spine. This is the postsynaptic spine. So this arrangement is such that whenever glutamate arrives at this metabotropic glutamate receptor, mGluR, immediately can trigger this response, and large amounts of 2-AG can be produced in a very small time, very small amount of time. So here is the 2-AG that is produced. I symbolize it like this. And what it does, it binds to the cannabinoid receptor, which not by chance is actually located exactly where is shown in this figure. The cannabinoid receptors sit in a sort of a ringlet that surrounds the synaptic bouton, just like this structures here, which we call the endocannabinoid signalosome, sit in a ringlet that constitutes the so-called perisynaptic region. So everything is very elegantly arranged for having large amounts of 2-AG produced, travel back to the presynaptic bouton, find the cannabinoid receptor. So why is that useful? What is the point of that? It's a little bit complicated. Why do we need that? Well, we need that because what activation of cannabinoid receptor does, as shown here, is modulating the release of transmitter. So if you have a little bit too much glutamate that is released, the glutamate will spill over to the perisynaptic region, so this little ringlet, and around the synaptic zone, the active zone. It will spill over, cause the release of 2-AG, and 2-AG now will travel back, activate the cannabinoid receptor, and tell the presynaptic neuron, slow down. We are excited enough. So it's a very, very important mechanism for feedback from the postsynaptic to the presynaptic component of a synapse, and that feedback is essential to brain function and is widespread throughout the brain. So if we have a feedback system like this, we also must have a system to stop it. Otherwise, a signal that is not stopped is completely useless. And what is the stopping mechanism? The stopping mechanism is just as elegant as the rest of the system. There is an enzyme called monoacylglyceride lipase, or MGL, that is located in the presynaptic bouton, not in the postsynaptic spine, only in the presynaptic bouton. And actually 50% of the enzyme is bound to the membrane. The other 50% sits in the cytosol, so in the water-filled space inside the presynaptic bouton. It's important, relevant, that some MGL is in the membrane because 2-AG doesn't like water. 2-AG will not be in the intracellular space or in the extracellular space. 2-AG prefers to be on the membrane where other fats are located. So here you can see the orange-colored sticks here are molecules of 2-AG. And what happens is that they activate CB1 receptors, but you want this activation to be short and to the point, and then you want to get rid of the 2-AG. And here's where MGL comes into the picture. It will cleave 2-AG into something else called arachidonic acid, one molecule of arachidonic acid and one molecule of glycerol, which are then disappear very quickly. So this is, all in all, the mechanism of retrograde signaling, and it accounts for the vast majority of the effects of 2-AG in the brain. So what about anandamide? I told you before that anandamide is a local modulatory signal. What do I mean by that? I'll give you an example based on work done by a grad student, an MD, PhD student in my lab, Don Wei, who is now at UCLA. Don studied the role of anandamide in social behavior, in particular in social contact. What you see here, these two lovely creatures, they're cuddling, are two mammals just like us. And just like us, they like contact with their conspecifics. So members of their own species make them feel good. And this is what rats do, this is what mice do, and this is what we do. And that's actually one of the problems with the COVID-19, is that we have much fewer of these interactions, and this can have impact on mental health, but that's a topic for a different conversation. So social contact is very important for mammals and very rewarding for mammals. So animals that enjoy being in the presence of their conspecific. And why? Why do they have this feeling of pleasure? Well, there are neurons in a region of the brain called the hypothalamus, and particularly a sub-region called the paraventricular and supraoptic nuclei of the hypothalamus. And these hypothalamic nuclei contain large neurons that send their processes, their axons, to many parts of the brain. And they contain a peptide called oxytocin. And some of you, or maybe many of you, know about oxytocin as the love hormone, something along those kind of cutesy ways. But really, it's a peptide transmitter produced by magnocellular neurons in the PVN and the supraoptic nucleus that is released during social contact. So when an animal experiences social contact with a conspecific that is not threatening, of course, then oxytocin neurons become activated, and they release oxytocin in a region of the brain called the nucleus accumbens. And that's a very, very important region because it underpins also the effects of rewarding drugs. All rewarding behaviors somehow impinge on the nucleus accumbens, but also the effects of rewarding drugs like, for example, cocaine or heroin work on neurons of the nucleus accumbens. So what happens when oxytocin is released in the nucleus accumbens from oxytocinergic neurons in the hypothalamus? What happens is that Don Wei discovered that oxytocin activates specific receptors in cells of the nucleus accumbens, and these receptors cause the release of anandamide. And anandamide travels from the neuron where it is produced to other cells, neurons, probably other type of cells that also bear the cannabinoid receptor, the CB1 receptor, and it is the anandamide released by this action that mediates the effects of oxytocin. So if oxytocin is able to enhance the rewarding properties of social contact, it is due to its ability to release anandamide in this region, the nucleus accumbens. So just like 2-AG, anandamide is produced and deactivated in a stimulus-dependent manner, in an on-demand dependent manner, but you can see that its action is profoundly different. It's not going from postsynaptic to presynaptic, a distance of only a few microns. It's going around a distance of probably several millimeters, which for the brain is, of course, is a big space. So the formation of anandamide, is different. The idea is, the general concept is the same, but the specifics are different. So whereas 2-AG was produced by an enzyme called DGL, anandamide is produced by a different enzyme called NAPPLD. And again, it's when there is need for anandamide. So if you look for anandamide in a brain that is inactive, there is hardly ever inactivity in the brain, but let's say a brain that is under very little challenges, the levels of anandamide will be low, but you can increase it in different regions at different times through activation of this enzyme. Likewise, you can destroy it, and the enzyme that destroys anandamide is very, very important. It's called fatty acid hammer hydrolase, or FA. And again, just like in the case of 2-AG and MGL, the enzyme that destroyed 2-AG, FA destroys anandamide when anandamide is no longer needed. So what I gave you is a pretty broad overview, really broad overview of the endocannabinoid system. And there are two more things that I would like to address today. The first one is pretty quick and is just a statement of fact. that the endocannabinoid system, this system that we have seen is so widespread, so pervasive, and so important for so many different functions. This system is also the port of entry for THC in cannabis into our body. So again, let me go over the logic of this. For the production of the endocannabinoids, you need a lipid precursor in cell membranes, right? Different precursors for 2-AG and anandamide. Then there are enzymes, different enzymes for 2-AG and anandamide that cleave those lipid precursors and convert them and release biologically active endocannabinoids, anandamide and 2-AG. And then there are more enzymes that block the effects of anandamide and 2-AG by destroying them, by degrading them. Now, there are also some, and this is something I didn't tell you yet, but I will now, there are some enzymes that actually convert 2-AG and anandamide into biologically active molecules. But biologically active molecules, they don't work anymore on cannabinoid receptors, but on other receptors. It's almost like there is an avalanche, a cascade. 2-AG is produced, works on CB1, then another enzyme arrives and converts into something else that works on another receptor. This is a new field of biology. We don't understand it well, but it's something that needs to be looked into in the future. So THC can really pretty much affect all of these things because it affects cannabinoid receptors, and cannabinoid receptors can talk through their signal transduction mechanisms to these various aspects of endocannabinoid function. So when we talk about the effects of cannabis in the body, the effects of THC containing cannabis, of course, in the body. I'm not talking about CBD hemp. I'm talking about THC cannabis. Well, the first thing that we need to think is how does it interact with the endocannabinoid system? That is the first thing that we need to think. And now, some could be good, some could be not so good, because yeah, by mimicking the endocannabinoid system, THC in cannabis could be doing some useful things, for example, reducing pain, reducing nausea. But if it interferes too much with the endocannabinoid system that is so pervasive, is so important, we could also have problems, right? So that is something to keep in mind. The second point that I wanted to touch upon, and the last one of my talk, is a question. Is it possible to use the endocannabinoid system as a source for better medicines? The answer to this is still not out. The jury, as they say, is not out, but there are very good signals. So again, this is how the endocannabinoid system works. And if you think about it for a second, a way of leveraging all this interesting biology to generate new medications is blocking endocannabinoid degradation. Because if you block this process here, what you end up doing is having more biologically active endocannabinoids. But what did I tell you? I told you that the biologically active endocannabinoids are not present all the time. They're only produced under certain circumstances, and they're also only produced in specific places of the brain or elsewhere in the body. So if we block the degradation, what we're going to have is a much greater selectivity and safety than giving directly a cannabinoid receptor activator, which could work for some people, but is unlikely to work for everyone. So again, the idea here is that if we block the degradation of the endocannabinoids, they will accumulate and activate the receptor CB1 and also CB2 when there is need for this receptor to be activated, when the body knows that there is need for the receptor to be activated. So what can we do? Oh, by the way, this is an interesting thing. Cannabidiol, or CBD, which is the other main constituent of cannabis, may also work in part by blocking endocannabinoid degradation. There are many more things that cannabidiol does, but it also blocks one of the enzymes of endocannabinoid degradation, FA, and part of its actions have been connected to that. So how can we, what kind of inhibitors can we generate? We can generate, first of all, inhibitors of 2-AG degradation, and we could do so by targeting 3 different enzymes. MGL, which I talked to you about, is the presynaptic enzyme that converts 2-AG into arachidonic acid and glycerol, and is probably accounts for 90% of the degradation of 2-AG. But there are two additional enzymes I'd like just to mention. One is ABHD6 and the other is cyclooxygenase 2 or COX-2, which also contribute to the conversion of 2-AG into something else. And ABHD6 probably works postsynaptically, and COX-2 we don't really understand how it works, but it's interesting because it converts 2-AG into something also effective, but on a different target than the cannabinoid receptor. So without making this too complicated, here are some of the effects that in animal studies have been demonstrated for molecules, medications, potential medications that block 2-AG degradation. These are MGL inhibitors predominantly. They reduce activity, seizure activity in epileptic animals. They reduce pain behavior, anxiety, morphine and nicotine withdrawal, and they even normalize behavior in mice having this form of autism called Fragile X syndrome. There are some effects that are not mediated by the endocannabinoid system. We know that. There is a very strong cancer suppression and reduced neuroinflammation, which are due to separate effects. But importantly, when we look at the side effects of 2-AG degradation inhibitors or MGL inhibitors, we see right away that they have a relatively mild side effect profile. So they're not disrupting fundamentally the way the brain works. They cause some sedation, some cognitive deficits, but something that could be lived with if there was a substantial therapeutic advantage, right? So what about anandamide? Inhibitors of anandamide degradation are predominantly inhibitors of this enzyme, fatty acid hamatohydrolase, or FA. COX-2, which I showed before, may also play a role. But most of what we know is about, most of what we know is about FA. And FA inhibitors actually have a host of interesting functions, or effects rather. They reduce anxiety-like behavior. They reduce depression-like behavior very, very powerfully. That's an effect that occurs at very low doses of these compounds. They enhance social behaviors in models of autism spectrum disorder. They reduce nicotine addiction. So quite interesting molecules. And in fact, they've been shown to be effective in cannabis use disorder, which is addiction and dependence, and now being tested. By the way, also 2-AG degradation inhibitors, MGL inhibitors, have been tested in Tourette syndrome with some initial positive results. And so both now MGL inhibitors and FA inhibitors are in the very first stages of clinical testing. And we certainly hope, as researchers in this field, that these tests will lead to conclusive results in the next few years. The last thing I wanted to say about FA inhibitors is that they have a very mild side effect profile in animals and humans, even milder than the effect of MGL inhibitors. Okay, so before I close, just briefly, let's not forget that cannabinoid receptors don't do only good things, right? They are some things that they do are probably not so good. And so there is some reason to have compounds that block the cannabinoid receptor. These are called receptor antagonists, right? And they come into several different families. I won't really spend too much time, but I'd like to remind those of you who are interested, they are neutral antagonists, they are inverse agonists, and they are allosteric and bias modulators. So basically, a neutral antagonist is a compound that binds to the receptor and does nothing. It just stops other ligands from binding to the same receptor. An inverse agonist binds to the receptor and pushes the receptor in a direction that is opposite to the direction it would normally be pushed by an activator, right? So it's kind of a deactivator of the receptor. And finally, allosteric and biased modulators. They're very interesting because what they do, they actually they take advantage of the complexity of these receptors to bias, to push the receptor, or sway the receptor in a certain direction. So for example, if you have the CB1 activating two different signaling pathways, 1 and 2, and say signaling pathway 1 is really good and signaling pathway 2 is very bad, right? I'm very overly simplifying. But let's say that you have this situation. Now, a biased agonist could bind CB1 in such a way that it only goes to the bad one or the good one. So this is the idea of bias modulators of a receptor. And there is a whole host of companies and academic investigators are working on them with the idea that perhaps using an allosteric or biased modulator, we could offset some of the side effects that THC has. What have we learned of so many years? The first cannabinoid receptor antagonist was published in 1994. What have we learned in so many years about this way of interacting with the endocannabinoid system? A great deal. So we know that the inverse agonists, compound like rimonabant, for example, or taranabant, are effective in something called metabolic syndrome. Metabolic syndrome is the combination of obesity, hypertension, and hyperlipidemia that is so common in the Western world. And these compounds are actually very, very effective in reducing metabolic syndrome. But they also have CNS side effects, and that's not surprising because the roles that the endocannabinoid system plays in the CNS are so many, are so important. Particularly, I mentioned a role in anxiety, right? Stress. One of the CNS side effects of the receptor antagonist, CB1 receptor antagonist, is actually anxiety, depression, even suicidal ideation. So this is not a pathway that is going to go anywhere easily. Neutral antagonists may have the same effect in metabolic syndrome as the inverse agonists do, and they're under study. And finally, allosteric and biased modulators I mentioned above are also under study. So these are interesting areas for future research. So I really think that this is important. So I'd like just to briefly mention what we know about CB1 receptor blockade in metabolic syndrome. Again, obesity, diabetes, hypertension, hyperlipidemia. What have we learned? We learned that the effects that we have seen in human studies with CB1 antagonists were predicted by animal studies, and that's important. The neuropsychiatric side effects, anxiety, depressive symptoms, were also predicted by animal studies, and this should make us feel confident of the predictive value of our models as we test alternative approaches, for example, the bias modulators, but also antagonists of the CB1 receptor. They don't make it to the brain. They're designed so that they stay outside the brain and bind to the receptors in the periphery, like receptors in adipocytes or in hepatocytes, or in pancreatic cells, receptors that play a role in metabolic syndrome, but they don't play a role in the regulation of stress and anxiety. And just as a reminder, we can restrict cannabinoid receptors to this outside the CNS. There are ways of doing that, and these drugs are actually effective in obesity without CNS side effects. So with that, I will stop, and I'll take any questions if there
are. Thanks very much, Daniele. This was excellent, terrific overview of the system. We can talk hours about this. I would like to ask you 2, 3, or 4 questions. Perhaps I would like to start with the idea that there is an endocannabinoid deficiency syndrome. We know from other neurotransmitter syndrome that neurotransmitter systems are not always working as well in all people. And what do you think about this theory? We see that in certain diseases, for example, migraine or epilepsy or irritable bowel syndrome, there are some disturbances in the system. Perhaps you could elaborate a little bit on this.
Yes, absolutely. One thing that I think we need to appreciate about the endocannabinoid system is that it's an on-demand system, which means that it's crucial that the enzymes that produce the endocannabinoids and the enzymes that destroy the endocannabinoids are regulated appropriately. So it can happen, and it does apparently happen, there is some evidence that this is happening, that changes in these enzymes could lead to also changes in the ability of the body to produce the endocannabinoid at the right time, in the right way. And that could be called, if you like, an endocannabinoid deficiency syndrome, but I don't think we have really characterized such a syndrome. But I think we do have the right to expect that there will be lower levels of endocannabinoids or maybe higher levels of endocannabinoids in particular patient populations. For example, it is known, has been known for now many years, that in obesity, there are very high levels of circulating endocannabinoids as well as high levels of endocannabinoids in white adipose tissue. That is important because in that tissue, the endocannabinoids serve a thrift role, which means that they are involved in making sure that we incorporate as much energy as possible in our body depots. And of course, that can lead to obesity, and it's considered to be one of the components to obesity. We don't know what is the molecular underpinning of that particular situation, but it's a situation that needs to be considered. Another interesting situation is where you have an enzyme that lowers the levels of the endocannabinoids, or actually in some cases can heighten the levels of endocannabinoids. And that could be good in some ways if that enzyme is present in the brain. So there is an enzyme called FAH, fatty acid hydroxylase. I mentioned it several times in my presentation. And this enzyme has a mutant form. And this mutant form is a very specific mutant that causes the enzyme to be less active. In fact, the enzyme gets destroyed more quickly. So humans that carry that particular mutation are actually quite common, about 4 to 10% of the population, in some countries more than that. And when you have high levels of this enzyme, low levels of this enzyme, you have higher levels of anandamide. So it happens that people who have low levels of this enzyme and they have high levels of anandamide tend to be better at dealing with anxiety, tend to be better at dealing with depression. Also, there is one particular individual in Scotland who has no FAH at all, and because of that, her levels of anandamide are really high, and she feels very little pain, and she says she's always in a good mood. Along these lines, one of the mutations of FAH, the C385 mutation, is quite widespread throughout the world, and a study that I recommend you view if you haven't yet shows that folks who have higher levels of this mutation, more frequent mutation, particularly in Northern Europe, tend to be in a better mood than people who are in countries that have lower levels of the mutation, particularly in Southern Europe, which is kind of interesting. The higher levels of anandamide, the happier you are. Now, I'm not subscribing necessarily to this data, but it was published in a reputable journal, and I thought it was worthwhile mentioning.
This leads me to another question. And that is the idea that we can increase our endocannabinoid level, for example, by exercise or sports. And this can be anti-addictive. So you can produce your endocannabinoids actively by stressing your body. And we first thought that it's the endorphins, and now it comes out that their endocannabinoids may also be involved in this runner's high, and which is also used as treatment for addiction and depression.
That's a lovely question. Thank you for asking that. Many years ago, I think it was in '98, we published a paper showing that exercise, 1 hour running on a wheel, increases substantially the levels of anandamide in circulation. Other people have found similar things later on, and to the extent that anandamide can interact with the brain, which is not necessarily clear how, possibly through some peripheral mechanisms, that would contribute to the feeling of well-being that certain people have when they run. So that is certainly interesting. Also, levels of anandamide in various regions of the brain can be regulated by wheel running in mice and rats. And that is also thought to have an antidepressant effect. So again, we're looking at a system that is so pervasive and so widespread that is almost, playing a role in so many different physiological functions.
Okay, yeah. Then I have another question, which deals with differences between the action of THC and the action of endocannabinoids, because we often read, well, THC is acting similar to endocannabinoids in the central nervous system, or anandamide is the natural cannabinoid which is working as THC to the cannabinoid receptors. And but we know both that there are many differences, and one difference may be the duration of action, which is really very important. You told us that endocannabinoids are produced on demand and they act very shortly, while THC, if you take it, it lasts for hours. So perhaps you can elaborate a little bit on this difference between the plant cannabinoids and endocannabinoids.
Thank you. That's a great question, and I think you also gave, in a way, the answer. Basically, when with THC, obviously THC is a very hydrophobic molecule, which means that it really likes fat. It really likes to be in fatty organs. So when an individual uses THC, no matter how, smoking or eating it, there are a few organs in the body where it will go preferentially, and one of them is the brain. The brain is very fatty. So the fact that THC goes to the brain is important, and the fact that THC likes fat is important, because then it will linger, right? It will linger for many minutes and hours after a single administration. And while it is in the brain, it stays as THC, can be metabolized to something else called 11-hydroxy THC, which then becomes 11-carboxy THC. But 11-hydroxy THC is also biologically active. So while THC is in the brain, it will inevitably bind to and activate cannabinoid receptors. There is no inherent mechanism to get rid of it. But there are inherent mechanisms to get rid of the endocannabinoids, because they are signals, and as all signals, they must be stopped at one point, as I said before. So with an anandamide or 2-AG, we have different time courses. 2-AG is really fast and goes down very, very quickly in a matter of literally 1 or 2 minutes. With an anandamide, it can go on, anandamide can go on for several minutes, maybe even tens of minutes, in some cases maybe 1 or 2 hours. But so that's a time course that matches that of peptides in the body, like oxytocin. But we're still talking about very limited time frame, and most importantly, we're talking about specific locales, specific places in the brain, because you will not activate an anandamide, you will not produce an anandamide throughout the brain at any time during life. In fact, an anandamide gets produced in massive amounts after death. The brain starts making a ton of an anandamide. That's too late. We needed it before. But so for that reason, you have maybe an area, a focus area where there is a ton of an anandamide, but most of the brain doesn't have it. And THC instead has no choice. It has to go everywhere. So there is a timing component, which you mentioned, that is important, but it's also a localization component that is also important. Again, THC has no choice, must activate indiscriminately all cannabinoid receptors.
Okay, then as an expert in this field, I would like to ask you a last question. Sure. And this refers to clinical uses of your research and research of others. Currently, we are using cannabinoid receptor agonists from the plants, THC and CBD. And you also mentioned that there is research into using, for example, inhibiting the degradation of endocannabinoids, 2-AG and anandamide, perhaps other endocannabinoids. And there are first clinical studies coming out. And what is, from your perspective, currently the most promising, or what are the most promising medical conditions that could be treated with inhibitors of endocannabinoid degradation, so that they activate cannabinoid receptors at the site where they are needed?
It is a very, very important question, a difficult question to answer. We have been trying to go from the results of animal studies to potential clinical indications for the last now over 20 years. And that translation is very, very difficult, as you and the rest of the listeners know. We have made, however, a lot of progress. And so now I think for Phi inhibitors, so inhibitors of anandamide degradation, I think there is hope in areas of use disorders, used to be called addiction, but I think problematic use or use disorder is more accurate. Use disorders like cannabis use disorder, which is a real thing, is not as frequent as other forms of use disorder, but it's a real thing and problematic. But also in social anxiety, for example, and depression. These are all areas where, as we discussed, anandamide is thought to play a very important role in the CNS. There is also the possibility of using inhibitors of Pha for the treatment of pain if those inhibitors are restricted to the periphery of the body. This is something that I have not touched upon in my presentation, but people, including us, have shown that blocking anandamide degradation outside the CNS is actually much better at reducing pain and producing analgesia than blocking anandamide degradation throughout the body. Is an interesting result. So that's for anandamide degradation. For 2-AG degradation, there is also a lot of hope, and there are ongoing also in that area clinical studies in epilepsy, in Tourette syndrome. At least these are two that I'm quite aware of. But I'm sure that as clinical research progresses, new indications will also be identified. And these are really the two most advanced, clinically most advanced programs and most promising programs that I can think of.
So I would like to thank you very much. We will see what the next decades will get out of it. Well, see you soon. Bye-bye.
Thank you so much, Franny. Thank you for having me. Take care now.